Decomposition is a process carried out by physical processes and organisms. As such, there is temporal and spatial heterogeneity in decomposition. The predominant controls over decomposition change with time. Decomposition begins as soon as biomass is produced. For leaves, there's a whole host of bacteria, for example, that are on the surface of leaves that begin degrading leaves as soon as they're produced. Roots are the same things. There's a whole host of bacteria and fungi that begin to decompose the organic matter associated with live roots as soon as they're produced also. Now, the host of organisms associated with leaves, live leaves, is called the phyllosphere. And in addition to there being bacteria and fungi on the surface of leaves, there's also bacteria and fungi that colonize leaves, and these are considered endophytic. So phytic is plant, endo is within, so you have bacteria and fungi that are found within plants and within leaves. While leaves are still alive, you can find the hyphae of fungi within their tissues. And these are not all necessarily detrimental to the plant. A lot of these fungi can produce secondary compounds that alter the palatability of those leaves to consumers. Now, understanding the patterns of decomposition in space or time is generally done with experiments. So, for example, there are some experiments that we call litter bag studies that are used to understand how decomposition varies in space and time. And essentially what a litter bag study is it's a bag, generally a mesh, that you put litter in. These would be leaves that had recently been senesced, or roots, or other plant parts. And you weigh them, put them in a bag, put them out in the field, tag them so you know what biomass you have, and then collect them a certain period of time later. You can see in this situation that you have a relatively wide mesh, which allows organisms to come in and out of the litter bag itself. Other techniques are used for decomposition of other material. So litter you can get into a bag, but entire tree trunks or tree branches are harder to get into bags. And so there are studies that take logs and put them out in the field, and then, for example, periodically sample those logs to take a look at the patterns of decomposition. So this is a log decay study, and this is relatively recently. After these logs have been put out in the field, a disc was cut out, and they can take a look at the patterns of colonization by fungi, and then weigh this to take a look at how much carbon has been lost or other nutrients. Now, in general, when we describe decomposition, we generally consider it to be an exponential process. What that means is that more biomass, more mass is lost from a given amount of litter earlier than later. Being an exponential process, it's generally modeled with an exponential decay function. So you have the amount of litter at time t is determined by the amount of litter that was present initially, and then how much time has passed, and then a constant, this k constant, that describes the rate of mass loss over time. So here, when we're talking about litter and decomposition, we tend to think about mass loss because we're weighing it. And in that case, we're often thinking about carbon. And there might be other patterns that are associated with the change in concentrations of nitrogen or phosphorus. And I don't think we talk too much about this, but carbon is generally considered to be an exponential decay function. But something like nitrogen, you can actually have an increase in the nitrogen concentration or content, so the amount of nitrogen in litter over time, and then a decrease, because you can have importation of nitrogen into litter initially, and then it's lost over time. So these exponential decay functions are generally associated with mass loss or carbon. And slightly more complicated equations are used for thinking about nutrients. So this is what a typical exponential decay function looks like for mass loss. And in addition, the authors have shown some of the different classes of compounds that you would find in litter. So this is the exponential decay function right here, but you can see there's differential loss of materials. Initially, it's the cell-soluble material that's going to be lost. And we talked about how leaching is a very important part of decomposition. So all the soluble material is lost relatively quickly. and that's thought of as being kind of an initial phase of decomposition. After the most labile material has been lost either by decomposition or by leaching, it's the less recalcitrant organic matter, and that tends to be cellulose and hemicellulose. And you can see that initially there would be this much, and then there's less, and less over time. Now, what does increase are microbial products. And we said that there's chemical transformations. And there's very little microbial products initially, and then that increases over time, and then decreases after that. Lignin is a relatively recalcitrant substrate, and it doesn't vary quite as much. And it's usually in the later stages of decomposition that it decreases the most. And lastly, the authors show that you have some organic matter breakdown products like the humus, which actually increase also over time in the later stages of decomposition as there's more chemical transformation of lignins, for example. The x-axis scale here differs depending on what system you're looking at. Tropics, which tend to be warm, decomposition happens relatively quick, and within just a few years you could lose 80 to 90% of your organic matter. In contrast, in the Arctic, where it's a lot colder and microbial processes are much slower, it might take 100 years to get to the decomposition that you would see in the tropics after only 3 years. The differences in climate are important for generating differences in the ratio that we see of litter fall and forest floor carbon. So we're looking at differences in production of litter fall, for example, and then how much is left over at any one point in time. So forest floor biomass and above ground litter inputs are shown here for selected evergreen forests. The lines that are shown here essentially represent the ratio that you get between the amount of leaves that are produced and how much is sitting on the forest floor. In a tropical forest, production is high, but decomposition is high. And the ratio here ends up being relatively high because decomposition is so much faster. And so in that case, we don't have a lot of carbon that's sitting in the forest floor, but we do have a lot of production. On the opposite end of the spectrum, we get into temperate deciduous and then the semi-arid systems, so drier systems, where decomposition is really inhibited relative to production. The K's that are shown here are the decomposition constants from the exponential decay function. So if you have an exponential decay function and you have a K of 4, that's going to go down relatively quickly compared to a decomposition constant of 1 or 0.1 or even lower, which means that it's still an exponential decline, but it's a very slow exponential decline. In these semi-arid systems, for example, where decomposition is often inhibited by lack of moisture, you can have a relatively low decomposition and almost no loss of organic matter in the forest floor at any one point in time. In seasonal environments, microbial respiration often occurs over a longer time period and peaks later in the season than does plant growth. So we've talked about interannual patterns, for example, and how that decomposition changes over time. But there's differences in decomposition that happen within a season too. Some of this is associated with differences in temperature and moisture. But there's offsets between when plant material is produced, when plants use nutrients, for example, and some of the optimal times for microbial respiration. And these offsets are really associated with spring and autumn decomposition. So spring temperatures tend to warm up and microbes can be relatively active, and that's before a lot of the plants have started to grow. In addition, in a seasonal environment, autumn temperatures are still relatively high in the soil after they've cooled off in the air. So plant production and plant utilization of nutrients can be zero, but you can still have a lot of decomposition. The reason this happens is that microbial respiration occurs over a broader range of temperatures than does plant processes. And as a result, those shoulder seasons, spring and autumn, for example, are warm enough for substantial microbial respiration, while plants are inhibited by those temperatures. In addition, soils are buffered from temperature extremes, so it can be very cold in the air, but those soils can still be relatively warm, especially in the autumn. And that's number 3, is that soil temperatures lag behind air temperatures. So air temperatures can be warm in the spring and cooler on the soil, but when autumn comes around, those soil temperatures can still be relatively warm. Air temperatures can drop relatively quickly. Now there's temporal patterns in decomposition, and there's also vertical patterns of decomposition that happen within the soil. Decomposition is not the same at the soil surface as it is 10, 50, or 100 centimeters below the soil surface. Most decomposition occurs near the soil surface, and this is where litter inputs are concentrated. And so it makes sense that if you have most of your litter being produced at the surface, that this is where most of that decomposition is going to happen. And as you go down through the soil profile, there will be less and less decomposition because there's less inputs in these places, for example. But often the carbon that's down here is relatively recalcitrant because it's been processed a lot. So that soil surface is important because it's the site of leaf inputs. Roots in general follow litter. And so looking at the soil surface, most of our roots are going to be produced where most of the decomposition happens. And so most of the root biomass is up near the top, and as you go down, you'll have less and less biomass. That being said, half of the soil carbon is generally found below 20 centimeters on average. And if you take a look at the pattern of where roots are in the soil, as opposed to soil carbon, you find some similar patterns, but with important offsets. So on the x-axis here, this is soil depth, and it goes down to 100 centimeters. These data here are globally average depth profiles of soil organic matter and roots in the top meter of soil. So if we say, on average, let's take a look at a soil from around the world, how much of the carbon pool, and again this is scaled to 100% being the most, how much of the carbon pool do you find at a given depth? Well, if you take a look at soil carbon, the pattern looks something like this. On shallower profiles, you'll have more root carbon than soil carbon. It also declines exponentially, but you can see that there's an offset. And by the time you get deep, you have a lower ratio of roots to soil carbon. Now, part of that reason that roots don't follow soil carbon quite so tightly as you go down through a soil profile is that the carbon that's found at depths tends to be more recalcitrant. That means it's harder to break down. You'll have less decomposition of a unit of carbon at depth that you do in relatively shallow soils. And therefore, it's probably not as advantageous to produce roots at depth for the purposes of acquiring nutrients as they are in relatively shallow soils.